Hello, so today we're going to be discussing the steps involved in DNA replication. So here we have a strand of double-stranded DNA molecule. And so this is not entirely accurate. It isn't helical or anything, but for simplicity's sake, we will be using this as our model. Now, so the first step involved in DNA replication is to form a replication bubble. Essentially, what that means is to separate the two strands of DNA, and this occurs through something called an origin of replication. So, an origin of replication is a short sequence that is recognized by certain molecules and proteins and enzymes, and it signals to them to separate the two strands. And so, here we have the two strands separated, forming a bubble. And with this bubble comes along two forks. So we have a replication fork here and a replication fork here. And the reason why it's called a replication fork is because replication cannot persist unless the two strands are separated, thus elongating the replication bubble. And so you could say that they're quite literally a fork in the road. All right, now this is what the replication will kind of look like and what we'll eventually see when we look at it broadly. And so what happens when the two strands are separated is one strand will act as the template strand. So here we'll take this 5' to 3' DNA strand as our template strand. And so our DNA will be then synthesized using this as the template strand, and it will base pair complementary to the template strand. And by bidirectional, I don't mean that one will be synthesized like this and one will be synthesized like this. It's just that one strand, the entire DNA strand will be replicated from this one bubble. So one will be replicated from here all the way down to here. And of course, we can't forget the remaining DNA. So we have to replicate this portion too. And so this part of the bubble will be replicated, and so will this be. And so that's what I mean by bidirectional. Not necessarily by the direction it's going to be replicated. And so how is it actually replicated? So first we have an enzyme called helicase, which unwinds the two DNA strands, causing this replication bubble. And so of course, when the two DNA strands are separated, there's always a potential that the two DNA strands can reattach to each other. And so we have something called single strand binding proteins that prevent that from happening. Now I want you to take a look here. As helicase unwinds, you'll notice that the helical structure of the DNA looks like this. The strands are not intertwined as much. It's not pushed down. There's not much tension, essentially. However, as helicase continues to unwind the DNA, you'll notice that these helical shapes starts becoming more concentrated towards the end. It becomes concentrated and there's tension that causes it to not be easily unwound by helicase. And so that's why we have an enzyme called topoisomerase, which relieves this stress. And essentially what it does is it breaks the two DNA molecules, it swivels them and reattaches them. So you'll notice it goes from here, breaks apart these two DNAs, swivels them so that they're sort of flat and they're not intertwined, and then it reconnects them like this. And so as topoisomerase does its thing, you'll notice that there aren't as many intertwining, and so the tension is not as prominent. And so this is what it looks like broadly. We have the single strand binding proteins, the helicase, which separates the two strands, and we have topoisomerase, which relieves that tension. But how is DNA actually synthesized? How is it actually replicated? We have kind of the setup to it, but we need to now go into how it's actually done. And so first, we have to establish the fact that DNA can't be synthesized necessarily out of thin air. It needs to start off somewhere. And so that's where primase comes in. Primase creates a primer, which is consisting of RNA molecules. And so it kind of So this primer becomes a platform for which DNA nucleotides can then be added to, and DNA can kind of go on from there. So after the primer is completed, we have DNA polymerase 3, which then synthesizes it. And notice here that we know that the DNA template strand is 5' to 3'. And so given that DNA is anti-parallel, this must go from 5' to 3'. And so it's now important to note that DNA polymerase can only add nucleotides on the 3' end. It can never add to the 5' end. So you'll never see DNA polymerase 3 going from this end, building off the nucleotides from this side of the primer, always this side. And so that's where we're going to talk about the leading and the lagging strands. So just kind of going back to what we discussed earlier, it's quote unquote bidirectional, in which this side must be synthesized, the left side of from the origin of replication must be synthesized, and so does the right side. And so we previously went over the left side here, where it's pretty easy, 5' to 3', there's nothing hindering it. The bubble elongates from this way, so it can just continue building its nucleotides, no problem, very easy. However, when we go into the right side of the replication bubble, notice here that we can only synthesize from 5' to 3', only on the left side of the primer. But the bubble opens up like this, so as it continues, how are we going to synthesize? We can't move to the right, only to the left. And so you'll notice that we have to create something called fragments, Okazaki fragments. And so this is what it kind of looks like, where a primer will be synthesized here. DNA polymerase 3 will then synthesize its nucleotides up until when it's done, and another primer will then be synthesized once the bubble has elongated far enough, and the same thing will happen. So it kind of can only synthesize to where the bubble has elongated. So it's dependent on that bubble or the function of helicase. And so like I said before, it creates Okazaki fragments. So this is what we're kind of looking at. We have the primer, we have the DNA nucleotides that DNA polymerase provided us with. It's still fragmented. There's no, solidification here. There's no connection. And so this is where DNA polymerase 1 will come into handy. So notice here how earlier the ones that were synthesizing these DNA nucleotides was called DNA polymerase 3. This one is called DNA polymerase 1. And so DNA polymerase 1 will go in and substitute the RNA nucleotides with DNA, because we can't have random RNA nucleotides in our DNA. DNA is just DNA, pure DNA. And so once it replaces all of the primers with DNA, we need to connect these fragments somehow. And ligase comes in and connects these fragments. You see here, it's still connecting them. And so ultimately, this will be our lagging strand. So just going back here, we have the leading strand, which continuously synthesizes, but on this side, because it can only move from 5' to 3', and it is dependent on the elongation of the replication bubble, it is done in fragments. So that's why it's lagging, because it takes longer because of the primers, the additional steps of the DNA polymerase 1, ligase, all that. So now we're going to be talking about telomeres. So you'll notice here how, go back, we have the leading strand here and the lagging strand here. So ultimately, once we reach the end of it, we can see that the leading strand completely synthesizes towards the end. It can, because it's leading. It goes in one direction and is able to do that. But for the lagging strand, the last end right here, so assuming that this is the end of the DNA, of the template DNA, we'll notice that there's still a primer here that has yet to be converted. And that's because DNA polymerase 1 cannot substitute these RNA primers into DNA. There's nothing to go off of. It needs to latch into something to do its things. And so because of that, this primer is not actually part of the DNA, it's RNA. And so it's eliminated. And so you'll notice that every single time we replicate, there's always going to be a primer that won't be synthesized, that won't be substituted back into DNA, and so essentially the DNA will shorten every single time. And so we have telomeres for a reason, which are just repeating subunits of nucleotides over and over and over again to account for the fact that they're reshortening. And so if there is shortening, those repeating nucleotides can then be used in that case, because it doesn't provide any significance. It's just repeating boring old subunits. That's basically the end of this short overview. I hope this helps, and see you.